Hey Akshat, how you doing? Hi Avi. Look at all of you rolling in. Hi Ronit. What's up, Arnav? Let's see, who else is here? Jasmita's here. Rayad. Hope I'm saying names right tonight. Hi Karunya. Saivi. Serena, good evening to you as well. Serena, I noticed that you subscribed to my channel today, so cool. Glad to have you with us. Michael, hello. Gondar, how you doing? It was nice meeting you a couple of weeks ago. I am just getting set up, everybody, so just give me just a moment. I love saying hi to you all. I know Mr. Jones is probably still going with his, but that's okay. We can have a little bit of an overlap. Who else is here tonight? Hi, Roma. Raindrop. All right. I would love to know everybody who is here and who's not one of my students. Who is your biology teacher? Drop me that line and tell me who your bio teacher is. I would love to know where you're from. Ajit. Mr. Gan. Akshad, you got Mr. Gan. Gan is the man. Ajit, you have Miss Chow, who's like an angel. I love Mrs. Chow. You know, Mrs. Chow did the flowers at my wedding. Did you know that? She's amazing. Mrs. Chow, when I first came to Monte Vista as a student teacher, she is best friends with my mentors. So she's awesome. Who else? Arya, I have an Arya. Arya, you got Ms. Lerner. Ms. Lerner is amazing. Did Ms. Lerner tell you that she and I had dinner together last week and it was awesome? Gondar, you have Lerner too, you know. Eden. Eden, is that how you say your name? Eden? That's a cool name. I love you guys' names. Such cool names. What's up, Nitin, my man? Ashley, you got Lerner. Saivi, you got Gan. All right. Hi, John. Good to see you. All right. Look at Ronin. Ronin wants us to get started. He's like, what's up, H-Dog? Let's go. Hi, Shida. Who else is here? Neil Kant. Am I saying that right? I apologize if I'm not. But 1 second. My neighbor just texted me. I'm going to text my neighbor back. All right. Let's see. Okay. So I wanted to actually start tonight's review session with I figured, you know what? Last time we got talking about some sort of stuff on the side. We went off on a tangent a little bit. So I wanted to start tonight's review session. Let me change my angle a little bit. I wanted to start tonight's review session by talking about food. And I wanted to specifically talk about a specific type of food and the food that I have brought to share with you tonight. I wish I could share it, but check this out. This thing is one of my favorite things on the planet. Anybody know what this is? This is a pomegranate. In Farsi, it's called anar. And this thing just, man, the way I do this, the way I eat this. So when I was a kid, check this out. I'll tell you a little story that used to make my dad mad. So pomegranates are not cheap. No matter where you are in the world, they're not cheap. So as a kid, I learned this from one of my older brothers. What he would do is he would basically like poke a hole in a pomegranate. Well, no, first he would like squish it all over. Just suck on that and basically just get all the juice out, which my dad thought was a total waste of money. So whenever I did that, I blamed my older brother, which is not okay. But what I like to do is I like to break it up. So cut it open with a knife, break it up, and then I put all the seeds in a bowl, right? And then from there, it's almost like cereal without the milk. So I'll grab a spoon in a large glass bowl or whatever, and I'll eat up. I'll be watching a show and eating it, and it's just amazing. I love pomegranates. Anybody else with me on that? I love pomegranates. All right. Let's see. Let's see what you all are saying. Ooh, lots of questions. Okay. We got to get started, don't we? All right. Rachel Chen, Learner told us that you guys are like goal buddies. Yeah, Rachel. So Learner and I are goal buddies. So the reason we went out to dinner is because, so something that you all may want to know about, maybe not, I don't know. I feel like our biology group, like we work our tails off. We really do. We're different in so many ways. We also have similarities, but one thing we all have in common is that we really care about our students a lot, and we want to make sure we give them our best. And I think, you know, in the teaching profession, sometimes you can give so much that you end up feeling kind of drained and exhausted. So we have come up with teacher goals, like personal goals. So last year, my goal was I wanted to, I forget what, I think it was like exercise 4 times a week or something. And for Ms. Lerner, she tends to stay at Monte Vista so late sometimes. So I challenged her to leave, I think, twice a week by like 4 o'clock or something. And she did. So I took her out to dinner last week, and we went to this awesome place that I would highly recommend called Nick's. And it's in Los Gatos. So if you haven't been there, I recommend it. All right, let's see. There's a fly in here. I'm not shooing anybody away. Okay, sorry if I did not say hi to you. I'm just going through real quick. Arshia, hi. Dia, hi. Shivali. Hi, Jenna. Nice hanging out with you again today. Jenna and I hung out a little bit. Good questions that you were asking, Jenna. All right, so John, what determines how a protein folds? So John, what determines it is you got to go through the steps of protein structure. So it's really kind of, man, there's so many things to it. So number one, the sequence of amino acids, like what makes it go in that sequence is the message they have from DNA, right? So DNA determines what sequence of amino acids are going to be put in. And then from there, you have your primary structure with the sequence of amino acids. And then you go into your secondary structure with alpha helix and beta pleated sheets, which is just kind of the way they fold. And then you go into tertiary, which is, you know, all that just kind of folds onto itself. And then you have quaternary, where you have 2 different proteins coming together. I hope that helps. Okay. So IV capillary action. I know that Ms. Detmer and I didn't really go over it a whole lot. So I didn't put anything on our test for it, but I can come back to you at the end of this. Try to, you know what I want you to do real quick, just type in capillary action, see what definition comes up. And if the definition doesn't make sense, I'd be happy to help you with it. All right, Gondar. Gondar, you a fan of pomegranates too? John, onion? Nope, not an, I do love onions. I do, but this is not an onion. All right. Gondar, pomegranate, yep. What are water's 5 properties? Okay, so Shivani, here's what I want you to know about water. My voice is cracked. What I want you to know is adhesion and cohesion. Those are 2 things that I want you to know. And then just know that water is polar. So I think maybe I can go grab a board and draw polarity with you all. So that's something I might do in a couple of minutes. But if you know adhesion, cohesion, and polarity, you should be good to go. Good job, Gondar. You did get it right. Pomegranate. You know, I just learned how to spell pomegranate. I learned it from my TAs in my sheltered bio class. Onion. Serena, is it a banana? You guys are goofballs. Okay. This thing is weird. Like I'm reading your comments and all of a sudden it goes down to the very bottom. Okay. So I'm going to try to get caught up. Let's see. Akshat. Oh, really? In Hindi, it's also anar? That's so interesting. I've learned that Farsi and Hindi have so much in common. A lot. Like we both have the word paneer, but in Farsi, it means cheese. All right. Hi, Komo. Let's see. Arnav, hello. Can you go over different biomolecules and different types of food? Yeah, so let's go over that real quick. Like proteins. You can find protein in, say, like meat. Meat has a lot of protein, or nuts, or dairy. Like egg whites, that's all protein. Carbohydrates, you know, when you think things like sugars, like monosaccharides or disaccharides, you're looking at fruits. When you're looking at longer chains like complex carbs, then you're looking at pasta, bread, rice, things like that. And then lastly, lipids, things like oil, butter. Remember, there are good lipids and lots of different things. Like nuts have lipids, avocados have lipids. Cool. Matches my shirt. Yes, it does. You know why I wore this shirt tonight? So I just got home, actually. I was visiting my mom, who has, I think I've told my classes that my mom had a series of 3 strokes 7 years ago. So she's disabled, she's paralyzed. So I go and visit her as often as I can. And then I got home, and on my way home, I was listening to the radio, and I found out the Niners beat the Raiders. Say what? So I was like, all right, I'm going to wear this shirt that I think actually, I forget, I think Mr. Jones got me this shirt, if I'm not mistaken. It's got Rice, Montana, and Lott on it, which they're all former Niner players. John, you've never tried a pomegranate? I am gonna have to bring it to class and share some with you. Pomegranates are awesome. All right. Are you eat it that way too, Eden? That's for me, that's the way to eat it. Yeah. Okay, what's healthier, unsaturated or saturated fats? Okay, good question, Ishida. So I would say, generally speaking, unsaturated is better. However, then we have to break unsaturated up into monounsaturated and polyunsaturated. And mono is much better for you. Monounsaturated fats are much better for you than polyunsaturated fats. We will dive deep into that when we do physiology next semester. We'll really get into that a lot more. So I know I just kind of covered the basics of it. What would happen to a cell placed in a hypotonic solution? Okay, Ria. So what would happen is the cell would basically start to swell. So if it's in a hypotonic solution, that means that the amount of water in the solution is higher, the concentration, than it is inside the cell. So water would rush into the cell, and the cell would swell, and possibly it would potentially burst. If that cell is a plant cell, then it has a much lesser chance of bursting, because plant cells have cell walls, and those are pretty rigid, and they keep the cell. It was one of the A-level question requirements. Got it. Okay, Shira. I hope I was able to answer your question. It swells. Yep, you're right. Unsaturated 4 times a week. Sophia, what do you mean 4 times a week? Oh, exercise? Sophia, check it out though. I know now my new goal this year is actually, hi Dakota, my new goal is 5 times a week. And my goal last year was just kind of just do something for exercise. This year, I'd like to lift weights and do some type of cardio that really gets me to sweat. So I've upped my goals. Hi, Stefan. How do shrunken red blood cells affect you? Good question. Okay, so basically, you want to make sure that your red blood cells are in an isotonic solution, and if they're starting to shrink, let's see, how deep do I want to get into this? I think what I would just want you to know is that if your red blood cells are put in a hypertonic solution, that they're going to shrivel, and that's not good. I kind of want to keep it simple. Okay. Viba, why are you saying Dia? Back-to-back streams. Yeah, we got 3 back-to-back streams tonight because we're awesome. Nick's, you don't have good memory. Why, John? Why don't you have good memories of Nick's? I've been there twice. The first time I went to Nick's, Ms. Lerner had actually got me a gift card. I had just gotten married, and my wife and I think my wife and I went to Nick's on that gift card and loved it. It's amazing. It's a little pricey, but it's amazing food. Now, could you go over how to identify monomers, polymers given in a diagram? Okay, so I would say it's really important to know their shapes. So I think, you know what? I'm gonna go see if I can find my board. I think it'll be helpful for you also. Give me a moment. I'm gonna disappear for a moment. I'll be right back. You can, here, look at my pomegranate while I'm gone. Okay, I'll be right back. Could you? Hi, everybody. I am back. I brought my board with me because I want to draw some stuff for you that I think will hopefully help you. We'll see. I brought a little board with me from Monte Vista, but the last year I was doing a study session in the backyard, not a study session, a review session in the backyard, and I left the board back there and it got rained on, so it's looking kind of interesting, but I'm going to try my best to kind of clean it up so that I can draw a few things for you all. All right, that's probably as good as it's going to get. Okay, so a few things that I want you to know. Number one, remember that for glucose, it's going to look like that. Let me make sure that I'm seeing it. It's going to look like that, okay? For amino acids are going to, let me see, I might try to draw this in a way where you can all see. So we're going to have okay, there's an amino acid. And what's really important about an amino acid is this R group right here. This is the one, excuse me, why am I saying, yes, no, I'm saying the right thing. This is the amine part. This is the acid part. I remember it as acids are cool. Okay. And then someone asked me last week, like, do you have an easy way of memorizing these? I don't. If you do, teach it to me because I don't have an easy way of memorizing it. Okay. And then the other thing that I want you to recognize is this. And this is a nucleotide. Okay. I call this a house with a garage and a pool. The P is for phosphate or pool. Here's your 5-carbon sugar. And then there's your nitrogen base right there. Okay. I hope that is helpful. I don't know if that was. All right. I'm going to go back to what we were talking about. Can you explain polarity? Yes, I can explain polarity for sure. I'm going to draw this for you all. I don't know if I did this in all classes because we kind of got to it last minute. But here, check this out. This is polarity. So you have oxygen. It's so weird drawing like this. Here's my O. And you have your 2 hydrogens. Okay? H2O. What's up, Kuchuk? What's up, Dakota? My dogs are whining for some reason. Okay, so there's my H2O. It looks like Mickey Mouse, right? Why? Why does it not look like this? How come it's not like that? Well, it is not like that for a reason. And the reason is because when you have oxygen, remember electrons are negative, right? Oxygen and hydrogens, they're sharing electrons, but it's sort of a partial sharing. So it's not exactly equal. So here's your, there's your hydrogens. Okay? Because it's not equal sharing, oxygen is taking more of that. So it's like if I gave you a cookie to share, but I only gave you a tiny bit of that cookie and I kept most of it for myself. So oxygen's being kind of negative. That's how you write partial negative. Your hydrogens are going to have partial positive charges. And this is an example of a polar molecule. Okay? That's the idea behind polarity. All right. Let's go back here to see what preguntas you all have. All right. You love the food. Yeah, John, the food's amazing, but I would love to hear sometime about your experience at Nixon. What happened? Can you go over eukaryotic versus prokaryotic? Yes, Arnav, I can. And I really want to because I feel like I didn't go over it enough in class. So eukaryotic versus prokaryotic. First of all, examples. So eukaryotic will be like plant cells or animal cells. Prokaryotic will be bacterial cells. So eukaryotic cells are much more complex and prokaryotic cells are less complex. Eukaryotic cells are generally multicellular. Prokaryotic cells are unicellular. And the biggest difference is that eukaryotic cells have a nucleus and their DNA is in their nucleus. Prokaryotic cells do not have a nucleus, which means their DNA is floating. I think that covers it. I hope that helps. All right. Eukaryotic versus prokaryotic. What are the properties of water? Sivy, I think we went over that just very briefly. The last A-level question, what does it mean the graph levels off? What it means when I say the graph levels off is this is what I mean. Let's see. I mean, here's your graph, right?
I can search for graph levels if this is what I mean on your iPhone.
Isn't that crazy? I wasn't talking to you, Siri. Man, this is weird, guys. This happens to me often. Okay, so here's the peak. So as it's leveling off, this is usually what I'm talking about. Okay? And I'm not talking to you, Siri. Thank you, though. Thank you for trying to help me. I appreciate it. All right. What is the function of smooth ER? Smooth ER really has to do with synthesizing lipids. Can you explain which of the fats would be healthier? I think we went over that one already, Ishida. Monounsaturated would be healthiest. We'll talk about that more next semester, but that's all you need to know for now. How does protein transporters function? What do you mean, Dina? What do you mean by how do they function? Like what goes through them? Do you mean like, I think, so in my class, I really focused on protein channels versus protein pumps. And so protein channels basically help with things like facilitated diffusion, which doesn't require energy, whereas in protein pumps, they actually need to put in energy because it's going from a low concentration to a high concentration. How does the structure of the plasma membrane allow it to be selectively permeable? Because you have the phospholipid bilayer. It's got two layers. It's sort of constantly doing this, right? And because of its structure of having proteins in it, certain things can go through, certain things cannot go through, depending on polarity, depending on size. So it allows some things through, but not everything. And that's what selectively permeable means. Okay. Ishida, because I is confuzzled. That's awesome. Could you explain a gradient? Yeah, a gradient is basically, man, this board is coming in handy. Are you all digging this board or am I just kind of, is it just me? So a gradient is like, imagine if, here, I'm gonna make sure that you can see this. So imagine if there's some kind of a membrane in between and I have a lot of something on one side and I don't have much of it on the other side. So that would be a gradient. Okay, Roma, I hope that is helpful. Tanish, I asked Mr. Jones, but I did not get a straight answer. What's up with Jones, guys? Seriously, what's with that dude? So I wanted to ask you, what is the difference of hypoisotonic solutions? Okay, so I think if you asked any of my students, they would probably give you an awesome answer, probably more awesome than me. But I will explain it to you this way. This is what you need to know. If you put a cell, let's say that cell is 97% water, okay? If you put that cell in a solution, that's my beaker, my solution has 96% water. I want you to, and sometimes I could say something like, instead of saying 97% water, I could be like 3% NaCl, sodium chloride. Or I could say 4% glucose. It doesn't matter. Don't pay attention to any of the other stuff that I say. Pay attention to how much water there is, okay? So in a situation like this, where do you have more water? Inside the cell, right? So you have 97 in there, 96 in here. So what happens is it goes from an area of higher concentration or high concentration to an area of low concentration. So it goes from 97 to 96. So number one, water comes from the cell into the solution. So it leaves the cell. As it's doing that, what's happening to the cell? The cell's probably going to start to shrivel a little bit. And when that happens, we call that a hypertonic solution. If the water was going this direction, it would be going from outside to the inside of the cell. So entering the cell would be swelling, okay? And it would be a hypotonic solution, like a hippo. And then lastly, if it's isotonic, that means you have the same amount of water, okay? Don't say water is not going anywhere. It's still moving in and out of the cell. All right, I hope that is helpful. Whose question was that? That was Tanish. Tanish, I hope that's helpful. I want to know how to pronounce this name, DHRUV. I would love to know how to pronounce it. Can you go over capillary action? So I didn't talk about capillary action in my class. Maybe if you have time at the end, I can go over it. But There are some of these that you can just kind of look up the answer to. And I know Jones did this earlier too, but if there's something that you can easily look up the answer, I might skip over it. I apologize because I really want to make sure I get to the deep stuff, especially because I don't have capillary action on the test. So you don't have to worry about it. Okay. Let's see. I lost my place. Where are we at? Where are we at, peeps? Oh my goodness. I lost my place big time. There's so much that I need to get caught up on, you guys. Where the heck am I? Whoop. Wow. Properties of water. Okay. What is the function? Okay, we got that one. What are some connections between protein synthesis and cell transport? What? We haven't even gotten to protein synthesis yet, homies. I don't know. Are you asking that because you're just kind of, this is from Maria. Are you asking that because you are just interested, or do you feel like you need to know this for tomorrow? Because I haven't even gotten to protein synthesis yet. I mean, I'm happy to teach it to you, but I worry that we're going to lose a lot of people's attention because it's not something I've taught yet. Okay. I love that you're helping each other out too. I see that you're helping each other. So if you can answer someone's question, please do. I would love that. Includes meniscus. Okay, so yeah, the whole meniscus thing. So meniscus is this. I did this briefly, I think, in some classes, where if you have a test tube, okay, you have a test tube, and you pour water in it. Like, what does this do? This action? How come it's not just straight line? It does this because of adhesion, right? It's sticking to the sides of the glass. And so cohesion, they're all sticking to each other. Adhesion is when the water is sticking to the side of the glass. So, and this is called the meniscus, the fact that it's not a straight line. All right. Smoothie out produces lipids. Yep. I love it. You're helping each other out. Voice cracks are okay. Thank you, Sophia. I appreciate the support very much. I always feel weird when my voice cracks. Good, Viba. There's fiber. No, Viba, don't worry. Don't stress out. All you need to know is adhesion, cohesion, and polarity for water. Can you talk about activation energy, please? Yes, Shivani, I can talk about activation energy. I will talk about activation energy. Am I using my board too much? I don't know. I'm having fun with it. So check it out. Generally speaking, well, not generally speaking, this is true for all reactions. All reactions have activation energy. Activation energy is the amount of energy it takes to get a reaction started. It's like, I'm sitting on my stool right now. If I want to get up, it's going to take energy for me to get up. So the activation energy for, say, like this reaction is that much. That's how much energy you need to get it started. And then from there, it's downhill. You're good to go. What's really awesome about an enzyme is that enzymes lower the activation energy. Okay, so an example of it is, what was it? Carbonic anhydrase. And you can look up what carbonic anhydrase does. Basically, as far as its products, it can make, so the reaction that doesn't include carbonic anhydrase, which is an enzyme, by the way, because it has an ASE ending. It's a reaction that has to do with your blood and CO2 and whatnot. You get 600 products per minute without the enzyme. Actually, you know what? I think it's 200 a minute. And with the enzyme, with carbonic anhydrase, you get 600,000 products per second. That gives you an idea of just how awesome enzymes are, and that without them, life would not be possible. Our life, at least. Okay. Glitchy, yeah. Chloroplasts produce ATP, right? Chloroplasts do not produce ATP. No, we would be talking about mitochondria. Chloroplasts make sugar. Paneer is cheese? Viva, really? Same. See, my voice cracked again. What the heck's going on? Yeah, paneer is cheese in Farsi too. We pronounce it paneer. Persians kind of roll their R's a little bit. We are Iranian, we roll the R. All right, Arya, why is osmosis an example of facilitated osmosis? I wouldn't say it's an example of facilitated and simple diffusion necessarily. Osmosis is the movement of water from an area of high concentration to an area of low concentration across the membrane. Simple diffusion is the same thing, except it could apply to molecules and not water, and facilitated is the use of protein channels. So if you can keep those things separate just as long as you're able to understand all of them. What is adhesion, cohesion, and polarity? So Roma, I went over polarity. So if you back up a little bit, I drew it on my board. Adhesion is water molecules sticking to something different, molecules of a, let's say like to glass. Cohesion is water molecules sticking together. Paneer cheese is Hindi too, but more specifically, oh really? Cottage cheese? You know, I didn't know cottage cheese was a thing until I moved to the US. I really didn't. In Iran, there was one type of cheese, feta cheese. That's it. Does the chemical properties and location of the amino acids cause determine the folding? Yeah, the sequence of amino acids, John, that determines the folding. Do we need to know about capillary action for the test? No, Surya, you don't. Not for my class and Ms. D's. Would all foods have nucleic acid? Pretty much, Shira, pretty much. Dina, yes, you. What is surface tension? So surface tension, Hira, is basically, surface tension is the combination of cohesion and adhesion. So it's kind of like if you ever see a bug that's able to sort of sit on water and not drown, it's just kind of like hanging out. That's surface tension. What is a chloroplast membrane made of, Arya? I can get into my, I can try to find that out for you, but I don't remember what a chloroplast membrane is made of. Hi, Parth. It's turning into a language translation course. Gandhar, if you were in my class, you would see that it's a combination of a biology and Espanol course together. So thank you, Ishida. I think the situation with my mom is that I don't know that she's ever going to feel better. I think it's just trying to maintain as high quality of life as we can for her. So, you know, it's like our parents take such good care of us, and I feel like it's our duty to return the favor if that ever is the situation. Is it really glitchy for everyone? Yeah, it got glitchy for me for a little bit. Good looking. Oh, okay. We got some weird comments over here. This good looking honky dude. What is the deal? Let's go Niners. Yeah. Where's Safwan? I know Safwan is my Raiders buddy. Where's he at? Is he hiding tonight after his 34-3 loss? What does glycogen do? So glycogen, Karthi, glycogen is basically a stored form of carbohydrates in animals in their liver. Did you know Ronnie Lott lives in? Yes, legit. I've heard Ronnie Lott lives in Cupertino. I would, if I ever meet Ronnie Lott, I hope I have this shirt on me and I hope he signs it. Ronnie Lott is legit, you guys. Ronnie Lott basically was in a game and something happened. I think his finger got dislocated or something. And he actually chose to get his finger like cut off to play the rest of the game. I would never do that, but I think that's cool. Let's see. Stefan, what requirements do we have to meet for the A level to 4? So Stefan, for the A level, so Ms. Detmer and I tried to be really specific with the help of many of you awesome students who gave us feedback. We try to be as specific and and make them as guided as possible. So if you can just follow those directions, you're good. What are different types of protein and their functions? Shivali, different types of proteins. I'm trying to think of whether I need you to know anything about that or not. And I don't think that you do need to know about different types of proteins. I mean, there are some proteins that basically have to do with, yeah, I'm trying to think. I don't think I want you to focus on that, actually. I think you'll be fine without it. I know it was in our review packet, but don't worry about it. Kushal, wow. Niners won. Yeah. Does the nuclear envelope protect our DNA? Yeah, it does. Yeah, what? All right. Let's see. Give me 1 second, you all. All right, I am I just had to remove this one commenter who was making inappropriate comments. So sorry about that. Hira, what are some substances that can diffuse through the cell membrane? Is it only non-polar substances, or can any substance diffuse through? No, not any substance can diffuse through Hira. So only something. So yeah, um, yeah, no, you got it. And I'm not getting too much into it because I'm not gonna focus so much on like, hey, like this thing is non-polar, is it getting through or whatever. John, why is unsaturated healthier? I don't understand. Ishida. So I know that because I probably didn't get into it a whole lot, it's a little confusing, but I promise that I will get into it when we get there. I just don't want to confuse you more by talking about things that you don't need to know right now. I see it as like bio is this kind of gradual, we'll take gradual steps into mastery. And so we'll get there, I promise. Ria Rai, why are unsaturated foods more healthy? Yeah, so Ishida was asking the same thing. I think for right now, you just need to know that monounsaturated foods are healthier. Now, the one thing that I will tell you is that saturated lipids, because they're sort of flat like this, they can actually pile on top of each other, right? And that could ultimately, over decades, clog up your arteries. Whereas unsaturated lipids, because of that on top of each other. So that's something I will tell you. Can you explain enzyme concentration in short part? So enzyme concentration just has to do with like the amount of enzyme that you have in an experiment. Saturated has more cholesterol. Okay. Ishida, I think. So I had triglycerides as the monomer of lipids, but the other day we said in class that the monomer of lipids is a fatty acid. Yeah, I guess I don't know. I don't know what other teachers are accepting, but for me, if I asked you to tell me what the monomer of a lipid is, I'd want you to say something like either lipid doesn't have monomers or that lipid has subunits, and the subunits are glycerol and fatty acids, or you can just say it's triglycerides. I'll take any one of those. Check on Kuchuk. What's up, Kuchuk? Why are you whining, girl? Hold on 1 second, everybody. I'm gonna go open the door in case she needs to go outside and pee. You wanna go outside? All right. Thank you for your patience, everybody. Okay. How does decreased activation energy correlate to a faster reaction rate, Stefan? So, Stefan, if you're decreasing the activation energy, that means you don't have to put in as much. So it takes less time. Okay. Where's the function of the cytoskeleton to keep the shape of the cell together? Will studying the review packet be enough to prepare for the exam? Minsu, I would say no. Studying the packet will be good. It'll be nice. But in my class, I would expect my students to review their skeleton notes, to review like all the materials they got back. And then the study packet should be kind of a sort of a review or a debrief of it all. Parth Jones is streaming. That's good. I'm glad we got both of us going. What does each biomolecule do to the body? We need these biomolecules for maintaining life, right? So like we need proteins to build muscle. We need proteins for enzymes. We need carbs. Like even when you think about cell membrane, what does the cell membrane have? It has made of lipids. It's made up of proteins. It's made up of carbs because the glycoproteins. So we need them at cellular levels, but we also need them at bigger levels. Okay. Gondar, I'm sorry that it's lagging. I don't know what's going on. Yeah, mine's kind of unclear too. It's weird. I don't know what's going on tonight, but I didn't like it. What is the general function of proteins in the body, John? So, John, they do things like, we talked about enzymes, we talked about muscles, we talked about, I think, you can talk about sort of how they fight disease in our body. Do animal cells have vacuoles? John, so typically vacuoles are for plant cells only. Wait, I thought it was bad because of double bonds. No, the double bonds are actually good, Ishida. The double bonds in unsaturated lipids are good. Can you talk about the parts of the cell membrane? Yep, just did. Shivani, I hope that me just talking about it right now was good. Animal cells, vacuoles are smaller, but yes, I think. Can you explain chloroplasts are larger than ribosomes using the Arya? So it's not something we got into at all with the endosymbiotic theory. I think it's awesome that you guys did, but if there's time at the end of it, I can come back to it. Let's see. Turgor pressure. You know what I need to do? I think I'm falling behind too much. So I'm thinking maybe I'll try something new and I'll try to work my way backwards. That might be a terrible idea. Let's see. Explain the metabolism of carbohydrates and how they're built up and broken down, Hannah. So basically, with carbohydrates, we break them down into small pieces, such as glucose, and then we save those pieces. So we put them together in the form of glycogen, and we save those in our liver. All right. Keep going. What is a glycoprotein? Karthi, a glycoprotein is basically a carbohydrate chain that's hanging off of a protein channel, and the cell uses it as identification. Where's DNA located in a prokaryotic cell? If there's no nucleus, that's coming from Prasant. Prasant, the DNA is just floating basically if there's no nucleus. Parth, mine lacks 2, it reloads it. Yeah, I don't know what's going on with that. Sorry, everybody. I don't know how to fix it. All right. What does the rate of diffusion depend on? The rate of diffusion, Surya, depends on lots of things. It could have to do with temperature. It could have to do with concentration of different things on both sides of that membrane. Saturation point. Okay, someone's asking about, Alex, you're asking about saturation point. So the saturation point is the point at which every enzyme is occupied. Cool. Huh. Interesting. By the way, I heard Jones recently talk about this whole idea of saturation point, and he talked about, I think, a grocery store and how if you have, say, like 3 cashiers at Safeway, the cashiers would be enzymes and substrate would be the people at Safeway, right? So if you have, how can you get the most amount of people going through the checkout lines at Safeway? Well, you can bring in more people, but if you have 3 cashiers and they're occupied, that's as fast as they're going to be. But if you bring in more cashiers, so if you add more enzymes, typically it helps. All right, for the enzyme graph, it's substrate concentration on data. What is it called when the slope levels off? That's from Iswar. Iswar, I didn't give it a name in class. Can your dogs make a cameo, please, Shivali? Yeah, Shivali, let me see if I can call my dogs to make a cameo. Kuchuki, come here. Dakota, come here. Hi. All right, everybody. Here are Kuchuk and Dakota. Here's Dakota. Hey, buddy, come here. There's Dakota. Hi, buddy. And then there's Kuchuk. Do you see Kuchuk? Hi, Kuchuk. Want to come say hi? There's Kuchuk. Yep. Thanks for coming, puppies. All right. Let's see what else we got. Surya, the size, the surface area versus volume, I want you to think about it like this. Like if you have a ping pong ball, all the stuff you can touch, that surface area, the air inside of it, that's volume. So if you compare that to like a gigantic beach ball, right? Is surface area bigger? Yes. Is volume bigger? Yes. But which one increases at a faster rate? So that's the part you want to focus on. All right. And we got lots of spammers tonight. What is happening? That's a first. We're getting popular, peeps. That's what's happening. That is what is happening. Okay. Let's see. Where are we at? I'm gonna try to get. I wish there was an easier way for me to stay caught up. Okay. So yeah, I explained polarity a little earlier, so go back and you'll find it. John, do we need to know how to draw a single monomer? No, but you do need to know how to recognize them, but you don't need to know how to draw them. All right. Meenakshi, suppose a cell that is 18% glucose is placed in an 18% water solution. Does water move in or out of the cell? Here's what I would tell you. I would answer your question like this. You have to look at how much water there is inside the cell and how much water there is outside the cell. Wherever there's more water, that amount is going to rush the other direction. Water goes from higher concentration to lower concentration. Try doing that on your own. I was asking, why are lipids considered subunits? Oh, okay. So yeah, so lipids don't have monomers because monomers are repeating units, which means that they're identical to each other. It's like two Lego pieces that both be the same size, right? Whereas with subunits, they're not always the same size. Yeah, Alex, I got big dogs. I know they're each about 90-ish pounds. I love these dogs. They're like my kiddos. Limburg Vikings. I went to Limburg my freshman year, so yeah, Karina, go Vikings. Sanvi, love them doggos. Me too. Oh, okay. I should bring them to school, Gondar. Gondar, I would love to bring these dogs to school, honestly, because I think it'd be good for kids, for students. I call them kids. I think it'd be good for the dogs. But unfortunately, I can't because I think some people have allergies. And I think some people are afraid of dogs. And in a public school, you're technically not supposed to bring a dog to work, to school, unless it's like a seeing eye dog or, but yeah, I sometimes on weekends when I go to Monte Vista to do work, I actually bring my dogs with me and I play with them in the academic quad. And a few times I've run into students. Yeah, a lot of you are saying bring them to school. I would love to bring them to school. If I could bring my dogs to school every day, I would bring them every day, without a doubt. Can you explain the 1 to 2 to 1 rule? The 1 to 1 rule in carbohydrates. So Lavanya, that's a cool name. When you look at a molecule, if the amount of CHO, so carbon, hydrogen, oxygen, excuse me, oxygen, if it's 1 to 2 to 1, if the ratio is 1 to 2 to 1, you're looking at a carbohydrate. Jones is just playing Fortnite. Oh man, I love Jones. Y'all know Jones is one of my closest friends. Good guy. What are carbon molecules? Ishida, okay, you're helping each other. Good, I'm glad. Can you go over lock and key model? Okay, Jasmita, so I explained lock and key model as, every enzyme is very specific to the type of substrate that it can take. And then I heard from one of my TAs who's currently in AP Bio that Mrs. Fallon has told them not to memorize it as the lock and key model, so lock and key model. Okay. Hiranya, if you have two solutions, one with salt 10% and the other with substance 10%. So yeah, because all you're looking at is the amount of water. How much water is there? Let's see. Yep. How do biomolecules form? Bria, you're getting deep with these questions. I would say that some of it has to do with just kind of what directions your DNA is giving everything, and that's how it's formed. Being the subject of dynamic equilibrium. Yes, Ishida, we do dynamic equilibrium. Mr. H, will it sell biocards Be great review for the exam, Ronit. Yeah, I think so, Ronit. I think they're a pretty good review. I posted them in Google Classroom, but I wouldn't just rely on those. Everything's great review. Thanks, John. John, your support means a lot to me in class and on here. On here? In here? Whichever. How do we recognize separate monomers? If you can bring out the board and please show us, Nissa. How do we recognize different? So Nissa, I did this a little earlier where I drew all of them. So if you go back, you'll be able to see it. Let's see. Through what process is glycogen made? Hannah, I don't know the name of the process that glycogen is made. But if it's something that you want to learn about, you can look it up when this is all done. Because that's not something that I'm quizzing anybody on anyway. Okay. Let's see. What do we need to know mainly for the test? Surya, that is a pretty general question. You need to know all the stuff that I taught you in this unit. I know I'm answering a general question with a general answer, but all the worksheets you have, all the review forms you have, all of that. So stored form of glucose in animals. Yup. I love that you're helping each other out. Collaboration. John, surface area to volume review. So John, I would say as long as you know the basics of like which one increases at a faster rate, that's what I would kind of just want you to focus on. Just remember a few things too, like so the day that I was gone, the day I took I went on the field trip with the Avid students. I talked to Ms. Detmer beforehand, and we talked about like how do we talk about surface area to volume, and I talked about a few things like why is it that if I come home and the heater has been off, that I'll see my dog sleeping in a ball, right? And the reason for that is because they're trying to decrease the surface area, because the more surface area you have, the more exposed you are to the cold. Or like for my compost outside. So there was one day when I had bought some broccoli and then I forgot about it, and then my wife was gonna throw it out, and then she was like, "Oh, this goes in your compost bin, right?" And she grabs it and she just gave it to me, and I was like, "We got to chop it up." And the reason you'd have to chop up the broccoli is because you're exposing more surface area, so that my bacteria, my worms, it takes them much less time to actually decompose it. Okay. Gondar, please bring them. How cool would that be? Okay. Active and passive. So passive is basically transport, no energy, active energy, and you go from low to high. What does it say in our review packet? This is from Ria. What does it say in our review packet that hormones are proteins, but on the yellow flashcard it says a smoothie R. Okay. So hormones are made up of proteins. Or are they proteins or lipids? Oh, I see what you're saying. They can be both. Right. Where did he explain the difference? So adhesion, cohesion. So these are two things you can look up easily, but cohesion is water molecules sticking to each other. Adhesion is water sticking to something else. What? I don't know what that means, Jonathan. He taps to build instead of holding the mouse. I don't know what that means. For any osmosis problems, should we assume that everything besides water can't diffuse through the cell membrane? Yeah, that's fine. Why is starch considered as storing glucose for plants? Chains, and those chains is what we call starch. Serena, you're helping out. Thank you very much. Same as you, Parth. I appreciate it. Do enzymes make the things required to make biomolecules? Not necessarily, but they can. How does water polarity? So I don't know how to pronounce that name. Chaitra? I explained polarity a little bit ago. I drew it on my board, so go back and check it out. Can't believe that I still remember that. Hira, what are you remembering? What did I miss? You're saying you can't believe you still remember that? Right. Endocytosis is from low to high. Yeah. So endocytosis is basically when a cell wants to bring something in it. So endo, like enter. Oh, okay. Endothermic versus exothermic reactions. That's what you remember here. That's awesome. Uh, Prasant, are you telling Safwan to sit down because the Raiders lost? I can't brag too much though. The Niners are 2-7. The Raiders are what? 1-7. So we're not that much better than them, but the fact that we just beat them tonight 34-3 makes me really happy. In fact, when we're done with this, I'm actually gonna go watch some highlights from that game because I'm kind of bummed out I missed that one. Well, question related to the property of water cause formation of meniscus. Say what? Run it. You don't need to worry about that. You're getting super deep into it and you don't need to. Not at all. Viva, you don't need to know exothermic and endothermic. No. Soft one, it was 34-3, homie, not 31-3. Give us those 3 points. They matter to me right now. Gondar, are you a Niner fan too? Good. Me and you know what? I should have bet with Soft one today. Soft one, you and I should have bet today. I just honestly I went into it being like, well, I think we're not gonna do too well. But yeah, Soft one, we had a third string QB. We still be. By the way, what happened to Derek Carr? Did he get pulled out of the game? Because when I was listening on the radio, they were talking about how they brought in a different quarterback. So I want to know what happened. Okay. Yeah, I know, Saf. I know it's depressing. It's a depressing time to be a Raider fan or a Niner fan right now. Meenakshi, what does diffusion depend on? Diffusion depends on lots of different things. So we talked about like heat. We talked about the different molecules you have in there. We talked about how much of the molecules you have in there, how much water you have in there. We talked about the actual membrane. What does it mean for something to be polar? For something to be polar, it means that it's a molecule where there's partial or unequal, somewhat unequal sharing. So partial sharing of electrons. Remember, electrons are negative. So like in the case of water, oxygen is taking more of those electrons. So oxygen is going to have a partial negative charge. Do we need to know adhesion, cohesion, and polarity for the next test? Do you mean tomorrow's test? If so, yes. Derek Carter. I love that we're talking about the game right now. Garbanzo. Yeah, Gondar, 3 points do make all the difference. I agree. Let's see. Just wait, is this soft one? Is this really you? I don't understand. You're a Raider fan, but you're knocking the Raiders. Are you just really disappointed in them? Is that what's going on? Because you're kind of sounding like a. Are you a Niner fan? And you just told me you were a Raider fan. How do enzymes decrease? So Stefan, enzymes decrease activation. Oh, how do they do it? I would have to look that up for you, or get a chemistry teacher friend of mine to come and explain that. But you don't need to know. I love that you're asking this question, Stefan, but I'd have to do some research myself to find the answer. Okay. I have to leave in 2 minutes. What do I do? Ishida, why do you have to leave in 2 minutes? And also, why are you worried? You're going to do great tomorrow. Okay. Ishida, you can ask more questions. Did not cover polarity. Yeah, we covered polarity. South one, you're disappointed. I understand your disappointment. I'm disappointed in the Niners this year. I mean, so many injuries. What are you going to do, right? I'm just glad that I get to give Sophomon a hard time this whole year. You go over the difference between monosaccharides and polysaccharides and how to identify them. Monosaccharides are basically, it's just the monomer, right? Polysaccharides, you're talking about starch or glycogen. Other than glucose or some monosaccharides. So things like glucose, fructose, sucrose, things like that are mono and disaccharides. And usually they kind of just go in the same category. You draw a diagram to show how proteins are formed. Legit. So think about this. If you put several amino acids next to each other, what forms them together, the bonds are called peptide bonds. So ultimately, you can call a protein polypeptides, but you don't need to know how to draw that for us. Lockdown because my mom wants me to be safe. I don't know what that means, Ashita. Can you explain that? Okay, bye, Stefan. Bye, soft one. Is polarity one of the reasons for adhesion and cohesion? Yes, definitely. Over lock and key. So yeah, lock and key is basically how specific an enzyme is, and that you can't use any enzyme for any substrate. It's like a lock and key. You have to have the right key to go into the right lock. Video lecture and practiced. Yeah, I think Ronnie, I think you'll be fine, man. I hope. I think you've done your due diligence, and I think you'll be fine. Here, how do lysosomes look? You don't need to like really know what they look like, but it's basically like something that looks kind of roundish, ovalish, and have enzymes in them, and that's kind of it. Oh, Ishita's back. John, do we have to know how surface area volume affects diffusion, evaporation, and heat loss? It's not a bad idea, yeah. Yep. Wait, but do we need to know osmotic pressure? No, Jenna, we don't. What is a dimer? A dimer is 2 pieces, Avi. Monomer is 1 piece, dimer is 2 pieces. Who's saying bye? Meenakshi, bye. Vibha, is it the wrong analogy? No, it's I think it's right, but I heard from one of my Harshi from one of my back up. I heard from one of my TAs who is in AP Bio. Her name is Harshi. She was telling me that Mrs. Fallon wants us to like she said something to the AP Bio kids about how lock and key is not the right analogy. So I need to figure out why. Do we have to label cell parts on the test? Lavanya, not on my test. Basically, my mom put this weird app that shuts down. Oh, at 10, so I get enough sleep, or otherwise I'll be super. Okay, I understand. That's pretty smart and really cool of your mom, because she cares about your health. You mentioned that the AP Bio teacher doesn't use it. Yeah, but they don't use it, so I need to figure out like why. But for now, you can just know that, but I'll figure out why. On a test, will we have to roughly know where the organelles are located? No, you don't. Monomer proteins. Okay. Good night. Good night, Shivani. Gondar, good night. Okay. Looks like a lot of us are leaving. Cool. Well, I want to encourage you all to try the pomegranate. I started this review session with talking about pomegranates. So these things are amazing, seriously. One note though, if you're gonna eat a pomegranate, wear a shirt you don't care about, because if you spill any of it on your shirt, it's sometimes impossible to get it out. That's what I've learned. So pomegranate is what's making me happy nowadays. Let's see. Serena, why do you keep calling this a banana? I actually like it. I like that you're doing that. Yeah, pomegranates. No, cytoskeleton does not produce ribosomes. No, that's the what am I trying to say? What am I trying to say? I forget my train of thought. Shows you I'm tired. Korean, no problem. No problem. What? Nick's has good mushroom soup, Ashley? Interesting. I'm gonna have to try that. So Ashley, I got something that's called Nana's meatloaf when I was there, and it's freaking amazing. If you like meatloaf, wow. They also have this amazing steak, and I really like their bone marrow. If you've never tried bone marrow and you're interested in trying it, go to Nick's and try it. It's that place just, I feel like it blows my mind with how good it is. It's like it's pricey food, but it is so good. Is monounsaturated healthy? Yep, monounsaturated is healthy. You got the ribeye, John? How was it, John? I want to know how the ribeye was. Thank you, Parth. I appreciate it. I love having you all join me. So Yeah, I still see that you're answering each other's questions, so that's exciting. Sci-V, protein structure. Sure, Sci-V. Sci-V, you're a Mr. Gann student, right? I think I met you when I was subbing for Mr. Gann. Basically, its primary structure of proteins is the sequence of amino acids. Secondary structure is when that sequence, when that chain starts to kind of fold in different ways. You can fold in two ways, alpha helix or beta pleated sheet. Tertiary structure is when it actually folds onto each other to form a bulky protein. Quaternary or fourth level is when you get two proteins merging with each other. Yeah, hope that helps. Good night, Ronit. Anytime, buddy. No problem, Surya. Arshia, no problem. Jenna, what types of sugars are nucleic acids? There are no sugars in nucleic acids. Yeah, Ashley, you like their bone. Ashley, you are the first student who's told me that you like bone marrow. I have never heard a student say that they like bone marrow. I love bone marrow. The ribeye was awesome. That's cool, John. There's hydrophilic, hydrophobic. Hydrophilic is water loving. Hydrophobic is water hating. Why do I have fruit flies? Why the heck do I have fruit flies right now? I have two bananas hanging over there. See if you can see my two hanging bananas. See that? I think I'm getting fruit flies because of two bananas. Can't be my pomegranate. Okay. Let's see. And I lost where I was, everybody. I know we're kind of winding down. Maria, what does it say in our review packet? Hormones or proteins, but on the yellow flashcard. Did I answer that already? So Ria, it's because it could be made of both. John, not all proteins go through quaternary. That's correct. Not all proteins go through quaternary. Hannah, how do organisms increase surface area? Like if my dog is really hot and he wants to increase the surface area, like Dakota, the golden retriever, he'll just lay out like a person. If he's really cold, he'll get into a ball. Have similar stuff, but we don't all have the same test. What's the difference between cytoskeleton and cell membrane? Cytoskeleton is inside the cell and it holds up the shape of the cell. Cell membrane is supposed to be for protecting what comes in and out. Quaternary is optional. I guess you could say that, yeah. Jenna, that was on the review packet. Really, Jenna? I'm sorry if I put that on the review packet because that's a bad question. What is bulk flow? Has a different environment. Don't worry about bulk flow. I took it off because I didn't talk about it too much in class. Where are we at? Yep, Gondar, you got it right with proteins and lipids. Parth bananas are tasty. Yes, they are. Do we need to know motor proteins and macromolecules? No, you don't. Not for my class. Is pH acidic 1 to 6, neutral 7? Yes. Neil Kant, you are correct. Is pH optimal for different catalysts? Yes, every enzyme has a different optimal pH. It's not the same for all of them. On the OChem practice sheet, there was a green molecule, then a blue one, and then a green one. It's a dimer. I don't know which one you're talking about, Hira. I'd have to see it. Ashley, you like the duck confit? Ashley, I feel like you have a very what's the word I'm looking for? Your palate is graduated, for lack of a better word. We're gonna have to talk food sometime, Ashley. Ashley, come visit me sometime so we can talk food. To reload. Do we need to know where organelles are in the cell? No, you don't. That is not the case. Dang, still 72 people watching. What are you all doing up at 10 p.m., homies? Why aren't you all in bed and like doing stuff that teenagers should do to enjoy life instead of hanging out with a bio teacher talking about bio and pomegranates? That's what I meant, Ajit. Palate is refined. That's the word I was looking for. Refined. Ajit, thank you for having my back. I appreciate it. If you happen to get a bad grade on the quiz, then up to a percent can be retaken. So in my class, it's up to a C minus. But Dina, I don't want you to think that way, please. I want you to think you're going to go into this thing and just kick butt. So don't start to think negatively, because really there's been research done that shows that if you go into a test believing in yourself and really thinking that you're going to do well, that you actually do better. I think it's by 10%. Surya aquaporins are basically like protein channels specifically for water. We're up at 10 studying for bio. Yup. 50 now. Okay. Thanks, Bart. Thanks for keeping count for me. Bye, Hira. No problem, Lavanya. You all are. I can't talk anymore. It's getting late. You are all amazing. I appreciate every single one of you. Ignore my bad explanations. Ishida, I want you to ignore my bad explanations too. Okay. Roma, we didn't talk about osmotic pressure too much, but it's this idea that there's so much water pressure that it's going to force water one way or another. So just know that, but don't worry about it. Avi, my favorite soccer team, just local, would be the Earthquakes. I love watching national teams, so I'm a huge fan of the U.S. soccer teams, especially women's team, because I think they're legit. I love the Iranian national team. And then I just like good soccer. I think if I were to pick a team from EPL, from the Premier League, I'd probably pick Liverpool. My wife is a huge Manchester United fan. And yeah, I used to be a Swansea fan, but they got relegated, which sucks. And I don't think they're coming back up anytime soon. Nissa, the reason we're here is because we're stressed about the test and trying to study a lot. I understand, Nissa. I totally understand. But why are you stressed? That's what I want to know. Like, what is causing the stress? I guess I want to know the root of it. Do you feel like it's because you procrastinated? Do you feel like you didn't understand material? Like, what is it? What are glycoproteins again? So Arnav, glycoproteins are carbohydrate chains that hang off of protein channels. Yeah, Gondar, thank you very much for looking up answers for me. Serena, nice yellow pomegranates in the background. I want to meet Serena. Serena's got a good sense of humor. Might have missed this, but can you go over concentration of enzyme substrate? Yeah, Ashley. So basically, enzyme concentration, substrate concentration, you just have to know that you can't always add more substrate expecting the rate of reaction to continue to increase, because you can hit a saturation point at which every enzyme is occupied. Okay. How do cells increase surface area? They just continue to increase in size, but there's a point at which they just stop and they can't increase surface area anymore. What are lipids, Jayanti? What are lipids made up of again? Like the subunits? Oh, okay. So it's got 1 glycerol and 3 fatty acid chains, and it's called a triglyceride. What part of cell does osmosis need help from? What do you mean, Ajit? It's getting so late, my brain's having a hard time thinking. What part of the cell does osmosis need help from? I think just the membrane. I think that's where you're going with it. Does osmotic pressure only occur in plant cells? Good question. I don't think so. I don't think it's just in plant cells. Aquaporins, monomers of lipids. What are glycoproteins and its function? Ria, so glycoproteins, we just talked about it like a couple minutes ago, so go back and see that one. What about La Liga teams part? I would so in La Liga I follow players more than I do teams. So I'm a Messi fan, not a huge Ronaldo fan, even though he's not in La Liga anymore. Yeah, more of a fan of just kind of like, I used to be a huge Real Madrid fan when they had the original Ronaldo, when they had Zidane, when they had Roberto Carlos, all those guys. Proper grades, Ishida. But here's the thing, though. I think we need to finish off here. I'm going to read a couple more questions, and then I think I know how I want to finish off this live review session by talking about proper grades and expectations and things like that. No problem, Arnav. My pleasure. Oh, Viva has been confused. So Viva, here's the deal, though. I want you to come and see me or Ms. D, or whichever teacher, and get help, during tutorial, lunch, like all that stuff. I want to see you all more. It's Monta Vista. We care about good grades a lot. I know, Jenna. I know. I went to Monte Vista myself, as you know, so I get it. Okay, so check it out. Yeah, Rio, no problem. So here's what I want to talk about. This whole idea of Monte Vista and like expectations and doing well and all that stuff. Like, I get it. I went to Monte Vista. I know that a lot of you put a lot of pressure on yourselves because you want to do well. I know that there's also peer pressure. I know there's competition. I know there's pressure from parents and families and sometimes teachers. Like, I get it. There's a lot. But at the same time, I feel like I've learned in my life to never, ever, ever sacrifice your overall health for, like, especially long term, for something like grades. I know that grades ultimately help you with your next step, which is college. But I can tell you this, I had a group of avid students who graduated 2 years ago in 2016. And I had students who didn't have 4.0s. I had students who had a 3.0 or whatever, like all over the place. And they got into the schools that they wanted to get into. Like, yeah, if you have a 3.0, you can't get into Stanford or Berkeley or whatever. But I think honestly, the way that I learned to think about those colleges too is that you don't want to wear a Nike or Adidas sweater, even though it might be uncomfortable and not a good fit for you just because it says Nike or Adidas on it. Like, you want to wear a sweater that's comfortable for you, that fits you, that like you feel good in. And so I feel like we put so much pressure on ourselves for like this next step. We don't even know what the next step is, but I think at Monte Vista we've gotten to the point where we start to think, well, if I get a B on a test, then I've failed. I've heard students say, "Hey, I failed my chem test," and I'm like, "What'd you get?" They're like, C plus. C plus is not a fail. Like, I know it's maybe disappointing, but I feel like we need to be a little nicer to ourselves, because I know a lot of you tried really, really hard and can't beat yourself up just because you tried and you didn't get the score that you wanted. So, you know, there's my little spiel. I feel like in life we have to be kind to ourselves and do our best, and then just kind of let it happen and see where it gets us. Yeah, apart expectations from parents. I know that that's true, especially with a lot of parents who come from different places. Like my own parents have worked their butts off to get us here, and so obviously, parents who are immigrants expect a lot of their kids sometimes, and I get it. But I don't know. I'm just saying, look at studying and look at school. Sometimes, don't forget to look at the big picture. And the big picture is overall, like, the most important thing is your health. Your academic health is important too. But yeah, no problem for the pep talk. Let's see. Okay. Ajit, one last question. Ajit, one last question. Bye, Parth. Have a good night. Are ribosomes produced in the nucleus or the nucleolus? They're produced in the nucleolus. That's the word I was looking for earlier when I was having a brain fart moment. Thank you for bringing that up and capturing it off. It's getting so late. I don't even know what I'm saying anymore, but I want to draw one last thing for you all because you all are amazing. So I'm gonna write this here. Let me change this a little bit. All right. Good luck. Lots of love. You're all gonna do amazing on this test. Try to get some good sleep, everybody. Don't stay up too much later. It's already 10:20. Take good care of yourself. Your brain needs time to process, and it's process. There's a lot of processing that takes place when you're getting good sleep. Cool. All right, homies. Take good care. See you all mañana.